Hello and welcome to Cardiac Pharmacology, Adenosine. Adenosine is a naturally occurring organic compound that consists of the purine base adenine joined to a ribose sugar. The goal of this lesson is to explain how, as myocardial energy stores are depleted, for example as might occur during hypoxic or ischemic conditions, cellular adenosine levels rise and cause a favorable change in the balance of myocardial oxygen supply and demand. The nucleoside adenosine is a critical component of nucleic acids, and adenosine is found in virtually all forms of life. Adenosine is also a component of important enzymes, vitamin B12, and the nucleotides ATP, ADP, and AMP. Adenosine is a very short-acting but powerful vasodilator. The half-life of intravenously administered adenosine is only a few seconds. Adenosine is also known to have important inhibitory central nervous system effects. Adenosine plays an important role in stopping seizures, and adenosine has neuroprotective effects during conditions of cerebral ischemia and hypoxia. In the 1990s, I was an investigator in a human clinical trial that evaluated the use of adenosine administered by constant infusion to lower blood pressure in human surgical patients. In my lab, I was also studying the electrophysiologic effects of adenosine in a Langendorf isolated guinea pig heart model and also in an in vivo animal model. One interesting thing I noticed was that not only was adenosine a potent sedative, adenosine also appeared to have powerful anesthetic-sparing effects. Here's a paper that we published in the journal Anesthesiology that examined the additive effects of adenosine on halothane anesthesia. The physiologic effects of adenosine are mediated by membrane-bound, G-protein-coupled receptors. Unlike the G-proteins coupled to the beta-adrenergic receptor, the cardiac adenosine-coupled G-protein inhibits enzymatic activity. Therefore, in the heart, adenosine receptor stimulation inhibits the activity of the enzyme adenylyl cyclase and reduces cyclic AMP production. Here's one way to visualize this. Adenylyl cyclase is the enzymatic engine driving cyclic AMP production. Beta-adrenergic receptors are the accelerator for this engine, and adenosine receptors are the brakes. If portions of this figure are not familiar, I suggest you go back and review my earlier cardiac pharmacology videos that will provide important background information. Epinephrine or norepinephrine stimulate the cardiac beta receptor that works through a stimulatory G protein to enhance the enzymatic activity of adenylyl cyclase. ATP, adenosine triphosphate, provides the energy for this process and is converted by adenylyl cyclase to cyclic AMP. Cyclic AMP then activates protein kinase A, which phosphorylates calcium channels located on the cell membrane and also on the sarcoplasmic reticulum. This causes intracellular calcium levels to rise and the heart to beat more rapidly and more forcefully. Cyclic AMP has a high-energy phosphodiester bond that is broken down by the enzyme phosphodiesterase. Cyclic adenosine monophosphate is then converted to adenosine monophosphate, and adenosine monophosphate is recycled to ATP. ATP is also made from ADP, adenosine diphosphate, and from adenosine. ATP is a crucial source of energy for many cellular processes. ATP is made during cellular respiration by glycolysis in the Krebs cycle. ATP is also made by beta oxidation of fatty acids. These processes all require oxygen. Hypoxia and ischemia result in ATP depletion. This figure represents what occurs during hypoxia or myocardial ischemia. The nucleotides ATP, ADP, and AMP are broken down to adenosine. As intracellular adenosine levels rise, adenosine diffuses across the cell membrane. The nucleotide phosphate groups makes these compounds very polar and prevents them from diffusing across the hydrophobic cell membrane. Adenosine, on the other hand, is much less polar and is therefore able to diffuse across the lipophilic cell membrane. Therefore, as adenosine builds up within the cell, adenosine is able to cross the cell membrane and stimulate adenosine receptors. These G-sub-I coupled receptors then very efficiently preserve ATP by reducing the activity of adenylyl cyclase. In the cardiac muscle cell, force of contraction is reduced. In cardiac conduction cells, heart rate is decreased, as is the rate of electrical conduction. It is important to have a solid understanding of these processes. During conditions of energy depletion, adenosine levels build up within the cell and adenosine diffuses across the cell membrane. Once outside the cell, adenosine stimulates receptors coupled to inhibitory G proteins located on cardiac myocytes and on conduction cells, resulting in decreased force of contraction, decreased heart rate, and decreased rate of electrical conduction. These processes each conserve ATP and lead to reduction of myocardial oxygen demand. As we've previously noted, adenosine is also a powerful vasodilator. Extracellular adenosine increases regional blood flow to ischemic tissue. However, because adenosine is rapidly removed from blood, these vasodilating effects are limited to nearby areas. Adenosine only increases regional blood flow to nearby tissue. Therefore, during conditions of energy depletion, adenosine is able to increase myocardial oxygen supply while decreasing oxygen demand. Therapeutically, adenosine is classified as a Class 5 antiarrhythmic agent and indicated for the conversion of SVT, supraventricular tachycardia. Adenosine is also used as an adjunct in cardiac nuclear stress test in patients who have limited ability to exercise. Dipyridamole, persantin, is a drug that inhibits movement of adenosine from plasma into red blood cells and endothelial cells. In this way, dipyridamole enhances the actions of adenosine. Naturally occurring methylxanthines include caffeine from coffee, theophylline from tea, and theobromine found in chocolate. These drugs are adenosine receptor antagonists. Methylxanthines competitively antagonize adenosine. Notice the chemical similarities between caffeine and adenosine. Because of the structural similarities, caffeine is able to bind to adenosine receptors. However, caffeine has no intrinsic activity or efficacy. It therefore competitively blocks adenosine receptors. By blocking the inhibitory effects of adenosine, caffeine is able to produce CNS and cardiac stimulant effects. This brings us to the end of this video. Please leave your comments below. I would like to again remind you to please support these videos by subscribing to this channel and giving these videos a thumbs up. Thank you.